Good afternoon, Denise, and welcome to your presentation for the California Master Beekeeper Program Master Capstone. And we look forward to hearing all the great work that you've done over the last 2 years. And it's going to be around 40 minutes of discussion. At the end, we'll go over a few questions. And we thank you for all the work you did and all the service in sharing all the work you did with all the other members and the general public. And so with that, I'll hand it over to Denise, and feel free to start the presentation when you're ready.
Great, thank you, Kian. And just make sure your volume is up for my first two videos. You want to hear what's going on, but then you can turn it down after that. So thank you so much, California Master Beekeeping Program. So my master capstone is which plants in San Diego County during drought conditions are providing copious amounts of pollen to our foraging honeybees, and what nutrition are they providing per analysis, including crude protein percentage, amino acids, and sterols. So I actually went back and added during drought conditions, because if you look at the historical county of San Diego with the drought, primarily it's more so in drought than it's not. We just happen to have the last 2 good years where we were not in drought. But when my project of collection was actually the year of 2022 here. So if you look at between 2022 and 2023, where my arrow is, it definitely was in the D1 and D2, which is the moderate drought and the severe drought. And that's important because these plants, even during drought, are providing copious amounts of pollen to all of our insects. And so San Diego County has many microclimates. It's quite large. And this black circle here is where I did most of my pollen collection. And the red dot is where I'm located. So I'm about 12 miles, a stone's throw to the coast. And this coast has a different microclimate than I do 12 miles inland. I'm up to about 10 degrees difference. And then if you go even further towards Ramona, which I will be moving to, could be another 10 degrees difference. And then you go into Julian, into the lower mountains, and then you can go all the way to Mount Laguna, which is 6,000 feet in elevation. And then on top of that, San Diego County still goes into Anza Borrego Desert State Park, which again is desert conditions. So within the San Diego County, we have many, many microclimates. But the plants that I did collect in this roughly 6 zip code area here, that's black, a lot of these plants are also found in other parts of the county. And I do notice that exactly on the coast that these plants will bloom about 2 to 3 to 4 weeks sooner than they bloom at my area. And then again, further out, they might be blooming a little further on down the calendar, but they are prevalent in a lot of part of the county of San Diego. So I just want to welcome to my office. So this project was out in the field. So I was not in front of a computer or had my head in the books all the time, but it was great to finally get out into the field and to actually be one in nature with all of our wonderful honeybees. So we're going to talk about pollen today. So what is pollen? So pollen is a powdery substance produced by seed plants. It consists of pollen grains, which produce the male gametes. And this picture depicts that each pollen grain has a very specific shape and size, but it has this hard coating that protects it during travel, whether that be via insect travel, via water travel, or via wind travel. Now, where is this pollen made? So the male part of the flowering plant is the stamen here on the left. You can see this full stamen, and then it has this yellow anther here. And pollen is indeed produced in the anther, and it usually contains about 4 pollen sacs, which are responsible for producing the pollen grains. And let's just move over to the right side of the flower. So this is the female part of the flower. This pistil, which includes the sticky stigma that accepts that pollen for it to travel down into the ovary and produce that seed. So which plants am I choosing and why? So I am choosing abundantly blooming plants providing copious amounts of pollen visually observed by honeybee collection during foraging. So it's pretty much my observation. So since I got into beekeeping at the end of 2018, I was always very interested in walking by all the shrubs and trees, and you could hear the hum and you could look up and you would see all the bees foraging. But then if you looked a little bit closer, if you looked on certain shrubs and trees, you would look at the bees, and the bees, most of the honeybees had pollen on their back legs, or most of the honeybees didn't have pollen. But nonetheless, these are the plants that I'm choosing because the honeybees are foraging and getting a lot of pollen from these particular plants. And again, my observations with my collection was the year of 2022, which is during moderate to severe drought categories per the U.S. Drought Monitor. So my observations were also backed up with the knowledge of sexing plants. So basically, these are just the basic sexings, includes the perfect, the dioecious, and the monoecious. There are other categories, but that are pretty complicated that we don't need to go into. But these are the reasons why particular plants are only giving up pollen and particular plants don't have pollen on them. And I will show you that in the next couple of slides. So when we look at a perfect flowered, or each flower is having both a functional male and both a functional female and male part. So on the left here, you can see the pistil here with a sticky stigma on the end, and then you can see all the stamens with all the anthers here. And this is our ironbark eucalyptus tree that are in my neighborhood, but they are just too tall for me to get pollen from. And this is a great source for my bees during the winter when it's blooming. They also have these pretty pink blooms over here as well. And then when we talk about dioecious, each plant is predominantly either a male or a female. And this is from Dr. Kim D. Coder, Professor of Tree Biology and Health Care and University Hill Fellow for Distinguished Outreach, Warnell School, the University of Georgia, Athens, Georgia. He was nice enough to be emailing me back and forth once I read some of his papers. And he said that when he talks about predominantly either a male or a female plant, it's about 90% or so. So when you look at the California pepper tree here, you can see this female tree on the right because it has all the berries. So that's predominantly a female plant. And then this tree on the left, right next to it, is predominantly a male plant. And it has no berries. And indeed, this was one of the pepper trees that I did collect some of my bees collecting pollen from it. And then you have monoecious. So this is a beautiful California lilac plant here, where separate male and female flowers on the same plant. So this cluster of flowers here, you can see these little yellow anthers up here on this particular little flower, but then on this flower right here, it doesn't have any. So the bee in the springtime, all the honeybees, they were copiously collecting a lot of pollen from this beautiful California lilac that we have in our area. And my resources. So I found this awesome book during a lot of my Google searches. It's called The Allergy Fighting Garden by Thomas Ogren, and it has this great opal scale in it. So I'll be referring to that when I'm talking about my plants. The Ogren Plant Allergy Scale, 1 to 10, 10 being the highest allergen / copious amounts of pollen. And it was so great because it was really hard to find the sexes of a lot of the plants that I was looking at. I also used the Beekeeping in Coastal California book by Jeremy Rose. He has a lot of plants in the back. Google searches, I attempted many library book checkouts that just fell flat for me. So this Allergy Fighting Garden book, Thomas Ogren is using it to instruct the public of what not to plant if you have a lot of allergies. But for a beekeeper, this is a great, perfect book to say what we do want to plant because we know that our bees need a lot of varied pollen sources. So how is pollen collected? So partly as bees fly, their bodies become positively charged with static electricity, such that when the bee lands on a flower that holds negatively charged pollen, the bee thus knocks the pollen from the delicate anthers, and some of the pollen particles stick to the static charged hair covering the bee's body. But that's not all. In this slow video, you can actually see this honeybee stroking her proboscis, and she's getting this nectar / saliva off of that. And so the honeybees collect pollen via the corbicula or moist collection. And they appear to do this during flight. So she's kind of hanging out in front of this wild artichoke flower that all the bees love. And also you see that native bee up there getting what she's needing. So we go to Ian Steele's book, Understanding Bee Anatomy. So the corbicula, or the pollen basket, is a concave shape of the outer surface of the tibia that can be seen there. And then the long hairs of the corbiculum house that. And so when the bees are using this nectar or saliva to gather up the pollen on the hairs of their body and shift it from one leg to the other, finally they put it into this back corbicula using that moist nectar and saliva. And you can see down in these pictures, this thorax of this bee is just full of hair. And if you see the close-up of one of the compound eyes, it is also full of hair. So really our honeybee has a lot of hair on its body to collect that pollen. And some native bees, such as this sweat bee, collect pollen made from the anthers of male flower parts via scopa, or dry collection. So she's rifling through this beautiful California lilac, and you'll notice on her back legs, there's no definite pellet there. It's just kind of this long source of the pollen that she's just packing on there, like this dry source of pollen. And then here the native bumblebee uses buzz pollination or sonication. So she grabs onto the anthers and then she buzzes it, and that pollen is released from the anthers and falls on her body, and then she can scoop it up and put it into her corbicula via moist collection. And this is done on plants that don't easily want to give up their pollen. Some of these plants include tomatoes, peppers, and cranberries. And so our honeybees are not big enough to do this buzz pollination or sonication. So it definitely needs the bigger bees, such as the bumblebee shown here. And here is just basic pollen collection and transfer. And you can see during mid-flight, she appears to be kind of gathering the pollen on all the hairs on her body. And she's looks like she's pushing it back to her corbiculas, back to her pollen baskets. And then boom, there's your pollen collection and transfer from plant to plant. So what is exactly in pollen that the honeybee needs for optimal growth and nutrition? So we're going to be looking at 3 different things: protein, amino acids, and sterols. And I have some research to back up all of this. So Dale Hill, animal nutrition consultant and primary developer of Dodon's AP23 pollen substitute, within his 40 years of experience, he states that there needs to be at least like 18 to 20% crude protein, that when ingested, these proteins are broken down to amino acids by digestive enzymes. And he states that the diversity of pollen sources is very important for bee foraging. No single pollen source meets all of the amino acid needs of honeybees. And these pollen is eaten by the nurse bees, and then it's assimilated and fed to not only the queen, but also to larvae. So now we're going to go into, so this is our crude protein percentages that we're needing, 18 to 20% generally. And now in 1953, amino acids, the 10 essential amino acids were found by de Groot. And he found it by an increase in the nitrogen content in dry weight was found to occur in about the first 5 days after emergence. Then a certain level is reached within which is constant. These changes run parallel with the development of the brood food glands, thus enabling the bees to secrete royal jelly, the food for the young larvae. In newly emerged workers, the saccules, or these pharyngeal glands, are small and pear-shaped. About after 6 days, due to intensive pollen consumption, the saccules are greatly enlarged and round off. So in this stage, the glands produce the royal jelly used for feeding the larva, the queen, and possibly the drone. So this is straight from 1953, and we do know that it is used for feeding the drones as well. And the following 10 amino acids were classified as essential for the growth of the honeybee: arginine, histidine, lysine, tryptophan, phenylalanine, methionine, threonine, leucine, isoleucine, and valine. And interesting enough, these same amino acids are required likewise for the optimal growth of the rat. And then if we move forward in time, Haydock in 1970 found out that there was a certain proportional ratio that was needed for honeybee health. and they found that these certain numbers here, arginine was 3.0, histidine 1.5, lysine 3.0, and so on. And he also found that at 12 hours after emergence, 50% or more of the workers have started eating pollen in small amounts. However, mass consumption begins when bees are 42 to 52 hours old and reaches a maximum when they are 5 days old. And the mandibles are used in crushing clumps of pollen or bee bread. So this was back in 1970. And I have asked two long-term scholar educators, and I asked them each, I go, well, do these young emergent bees, do they eat fresh pollen or do they eat bee bread? And one said bee bread and one said fresh pollen. So I'm sure that is probably a combination of both. And then we come fast forward to 2019 with Priyadarshini Chakrabarty, Hannah Lucas, and Ramesh Sagili, published in December 6, 2019, that there are a critical micronutrient called 24-methylene cholesterol, or 24-MC, that's needed, that these bees are needed at 0.5% survive the longest, and their head protein and abdominal lipid levels were also among the highest with this critical micronutrient. And one role of this micronutrient was important micronutrients that are essential for production of insect molting hormones. So we know that during the larval phase, they have every 24 hours is a different instar, so that larva grows for 24 hours and then it needs to shed that skin so it can grow again for the second 24 hours, because this is its feeding phase during the larval phase. And this micronutrient helps with that process. So this is why I'm analyzing our San Diego pollens, to determine the crude protein percent, the amino acids, and the sterols 24-MC present per pollen sample. So what are my collection parameters? So number one, abundant amounts of blooms on a large single plant / shrub, small tree, so during the full bloom period. Number two, visual honeybee collection of copious amounts of pollen. Number three, abundantly grown in San Diego County. Number four, it had to have been safe and accessible, public lands, non-sensitive areas. So this is an example of a full blooming shrub where you can not only see all the honeybees during their collection phase, but also you can hear them. So my collection equipment was pretty basic. I had my beekeeping boots, I had a backpack, some water, I had my butterfly net, and I had this rolling ice chest that I would put ice into, because once I collected those bees, I wanted to get them on ice and make sure that the pollen was preserved. And then I would bring it back home and I would immediately put everything in the freezer. And that was a humane way to euthanize those bees quickly so that I could take the pollen off of their legs later on. So that was my collection method. I got pretty good at this, and I originally had a headpiece on and long arms and everything, but the bees were pretty gentle when I was out collecting, so no problems there. But as you can see, the corbiculas are full of pollen. I made sure and only put one bee per collection tubule, and I did put air spouts in the tubules so that they could breathe while in them. And then also I put them on ice right away so their activity would slow down so they weren't too agitated with the collection. So this is my collection log, number of plant species for which pollen has been collected on the left, and then the list of the plants. I had sometimes up to 3 different addresses of where I was collecting from, and then I had date collected, time collected, I had temperature and humidity, I had climate conditions when collecting. And honestly, the lower the humidity, the better to preserve the pollen. And we did have a lot of low humidity times. We had even up to 18% humidity, 15% humidity when the Santa Ana winds were blowing. And then I had either wild or propagated. I tried to get as much wild as possible. And all the method of collection was by bee collection. And I had about 1,287 bees that were collected in about roughly 50 hours in the field. Okay, so I'm in the process of collecting or getting the pollen off of my frozen bees. So I just collected some arroyo willow and I have frozen my bees. Keep everything frozen, so the frozen bees are on ice. My collection tubule is on ice, and also the bees are on ice here. And I have these very special tools. I have a tweezers here and then a scalpel. And so if I grab the femur up here, I can just very easily pop the pollen pellet off. I flip the bee over and get that second pellet off. And then I can use my tweezers and pick up that pellet and drop it right into my collection tubule. And I thank these bees for being part of this research project. I try not to collect too many bees from one source, so because I know that their colony is close by. And here's a big pollen packet. Very pretty yellow pollen from the arroyo willow, which is a native tree. It has catkins. It's a willow tree, so it has catkins, and it's a dioecious tree, so it has a male tree or a female tree, and it's pretty easy to find the male trees. So I'm laughing because I said they were very special tools. The only reason they're special is because my husband had them in the garage. It's just a dissection kit, but he has every tool known to mankind in his garage for me to use, so it was great. So, and this is my washing methods after each specimen, just so that there was no cross-contamination from one pollen to the next. And so we're talking about my sources. I have 14 sources to talk about. The first source of the year was the African sumac, or the Rhus lancea. It was a dioecious evergreen tree, opals scale from 8 to 10, so it was very easy to pick out the male trees that didn't have any kind of berry on them. And they were in full bloom first part of January. And you can see the bee collecting here with, it's kind of like a mustard color pollen here. Now these blooms were very, very small, so I actually had to collect a little extra bees on this particular source just to get enough pollen, because I wasn't sure how many bees that I would need to collect per pollen source, but it was roughly averaged out to, I think, about 70 bees per pollen source to make sure I had enough pollen for analyzing. And then lemonade berry was the second source, Rhus integrifolia. It's a wild native, and it's, again, is a dioecious evergreen shrub to small tree, opals 8 to 10, found coastal and canyon slopes and flat foothills, blooming late January with a collection of 127 to 128. And you can see my, this is like a 6-foot pole here just to show you the what the size of the shrubs and trees are that I'm collecting from. And then over here, this is the flowers are just opening. You can see all the yellow anthers here. So this is predominantly a male shrub. And down here, you see a close-up to give you an idea what it looks like. And this is compared to the female tree, it's all pink in there. So you don't have any pollen in there at all. And then this female tree ends up with these berries here, hence called lemonade berry. My third source was the arroyo willow, what I showed you taking the pollen off the bee's legs. Salix lasiolepsis, wild native, found in riparian areas. And again, it's a dioecious deciduous tree, opals 10. And as you can see, these beautiful catkins have so much yellow pollen on them that the bees are happily collecting here. And then you can see the counterpart, the female catkin, very different looking, doesn't have any pollen whatsoever on it. My fourth source was the Sydney golden wattle that I showed you originally in the video of all the bees foraging. This is an Acacia longifolia. It's a perfect flowered evergreen shrub to tree from Australia. Opals 8 to 10. It's naturalized in all of our riparian areas here in Southern California, and this was blooming in February. And I will say, even though it's a perfect flowered evergreen shrub, every single bee that was foraging on this plant was indeed collecting pollen in February. My fifth one is the beautiful California lilac, Ceanothus tomentosus. It's our wild native, monoecious, evergreen shrub to tree, and it's an opals 4 to 5, but although it's only an opals 4 to 5, it's not closer to the 10. Again, every single honeybee that was foraging on these shrubs indeed had pollen in their corbiculas, and they were collecting pollen, and this was at the end of February into the early March. And then we have these beautiful coast sunflowers, or California brittlebush and Cilia californica. These are, again, wild, native, perfect flowered evergreen shrubs, opals 4. But I'll tell you, all the bees had this beautiful orange pollen on their legs that they were collecting from these flowers. And these are all over our parklands, hiking trails, and on all off the roadsides here. And I collected them in March. And then this was an unknown source to me that I just happened to go ahead and collect. It was the wild radish, or Raphanus rapinistrum. It's an annual perfect flowered invasive weed. So I was really happy to get some kind of invasive weed into this research. There's no scale in the book for this, but I'll tell you, even though it's a perfect flowered invasive weed, every single honeybee on these flowers were indeed collecting pollen. And it was really quite beautiful because it was a combination of white to light pink to darker pink in this meadow here. And number 8, I love this, my Mexican elderberry. I love this shrub, it's so gorgeous. This is Sambucus nigra. Occurs throughout the western US and Canada. This is a wild native. It's, again, a perfect flowered deciduous shrub to small tree. And the bees are just, again, every single bee on this shrub with the collection in May is collecting copious amounts of pollen. And you can see with my pointer here, there's about 5 stamens on every single flower, and they are getting these big balls of yellow pollen off of it. And then these blooms turn into these beautiful blueberries here afterwards. And then we have the California tree poppy, Rhamnea coulteri. It's a native, and again, it's a perfect flowered tall shrub, only in opals 4. But with the video, you can see every single blooming flower has roughly 5, 6, 7, 8 bees all in there collecting up their pollen and getting these big pollen packets on their back legs to take back to their cavities. And then we have the California pepper tree, Sinus schmoll, the dioecious tree, opals 10, because this tree is predominantly a male, and the bees are all over this collecting this kind of mustard colored pollen here. And indeed, this is not, California pepper tree is really not from California, it's from Peru. It just has naturalized here, and so they kind of changed it to the California pepper tree. But these trees are blooming all throughout the months in San Diego. And in fact, one tree that I collected on during this May session here also bloomed in the fall. And I also saw bees collecting pollen on it in the fall. So very important sources for our honeybees. Number 11, the honey myrtle, or pink melaleuca. So Melaleuca nasophilia, an Australian perfect flowered shrub to small tree. Opal 7, this is all over, propagated along city streets and highways, blooming early June. And again, you can see all of these stamens and all these anthers here. And so they're collecting quite a bit of pollen off of this perfect flowered shrub. And this pollen was a little bit different because it was very, very dry and very, very fine pollen when I was taking it off of the bee's legs. Number 12, beautiful laurel sumac, Melasma laurina. It's a wild native, dioecious evergreen shrub to small tree. Again, opals 8 to 10, because this shrub is predominantly a male, producing all of these yellow anthers here, getting all this beautiful yellow pollen. And then the crepe myrtle, perfect flowered deciduous tree, only in opals 5. But again, when I went to look at the bees, they were all collecting pollen, and you can see that each flower has a lot of different numbers of stamens. So there's a lot of pollen collection on these crepe myrtles. And this was in the month of July. And my last source is the coyote bush. And Baccharis pilularis, a wild native, dioecious evergreen shrub, opals 9. Again, it's dioecious, so each shrub is either a male or a female, and it's pretty easy. You can see all these little yellow anthers here that this bee is collecting from. This is found all over, and all over the roadsides and all over the place, and it blooms. in the fall. So this is a super important plant that when everything else is done, that this plant is still blooming, providing this pollen for them. So my one and only result was a protein percentage, and you can see all my plant species down here along the bottom line. And then if you look at the California lilac here, it had the most at 16.63% protein. And then you see the second one is another native, elderberry, and then the California tree poppy is a native, and then this other one, laurel sumac. So of the 4 top ones that had the most protein, they were natives. But I was kind of a little disappointed that none of my sources even came close to the 18 to 20% that Dr. Del Hill was referring to. So I'm really interested in getting my amino acid amounts to see how that all works out for the bees. So true conclusion, the study has simply showed us the numbers. But the diversity of pollen sources is very important for bee foraging. No single pollen source meets all of the amino acid needs of honeybees. And I have heard that many times and will most likely hear that many times to come. But however, more research is needed for the exact conclusions about how these local pollens fulfill the nutritional needs of honeybees as brought into a specific hive and consumed by the nurse bees. And luckily, Project APIS M's PAM mission is to fund research and efforts to improve honeybee health and vitality. Since 2006, they have infused over 10 million into honeybee research and 2.9 million into forage programs, resulting in science-driven resources for bees and beekeepers. And just to, I know you all know how important our honeybees are. This is from the Almond Board of California. Today, the state of California is the biggest producer of almonds, growing approximately 80% of the world's supply. Almonds are California's top agricultural export and largest tree nut crop in total dollar values and acreage. They also rank as the largest U.S. specialty crop export. And of course, almonds rely on honeybees for pollination, and the California almond community recognizes the essential role bees play in sustaining the global food supply. And so this is mainly about honeybees today, but I took a lot of pictures and some video of all of our pollinators are very important, and they are all also using all of these plants for their food sources. So I'm just gonna, if you want to just sit back and relax and just look through a little compilation of some pictures that I've been taking before I wrap up this talk. So I'd like to thank the California Master Beekeeper Program for educating me and giving me this opportunity. I'd like to thank Dr. Priya Chakrabarty Basu, Ramesh Sagili, team of Max Simon and Lauren Jennings for the USA Pollen Library Project. Without them, none of my pollens would be able to get analyzed. I would love to thank Dr. Dale Hill, Dr. Kim Coder. I had various other resources and my phone apps, iNaturalist and PlantNet. And of course, my husband for allowing me to do all this crazy beekeeping stuff. Questions and comments?
Thank you, Denise. Wow. Very clear, concise presentation, great graphics. And I mean, you have enough photos for a whole library there.
I know, and I kept getting more and more photos. I'm like, oh, I got to just show this.
Yeah, you should put it in like some photo library if you have your own personal website or something like that.
Should I stop sharing at this point?
Yes, and I think we can stop recording too.
Thank you.